Good afternoon. It is my pleasure to introduce today's keynote speaker, Professor Sergio Grinstein. Sergio completed his PhD in 1976 at the Centro de Investigación y Estudios Avanzados in Mexico. He then spent 2 years as a postdoctoral research fellow at the Hospital for Sick Children in Toronto, followed by a year in the Department of Biochemistry at the Federal Institute of Technology in Zurich. He is currently working at the Hospital for Sick Children in Toronto and has been a professor at the Department of Biochemistry at the University of Toronto since 1988. And funnily enough, a few years ago, Sergio was the first keynote speaker at the Montreal Light Microscopy course headed by Claire in 2010, where he did a really nice talk where he combined both the fundamental methods as well as advanced methods to investigate phagocytosis. So Professor Grinstein, welcome, and we look forward to your talk.
Thank you very much, especially Claire, for inviting me yet again. As you just heard, Claire invited me to be a speaker 15 years ago. She claims she still remembers part of my talk. If her memory were that good, she wouldn't have invited me, so don't trust her. What I'll try to do today is talk about a biological problem that in all certainty most of you don't give a damn about. Yet I'll try to convince you that regardless of the nature of the biological problem, the proper and intelligent application of imaging techniques can be incredibly powerful and essential to go from the tissue level to the cellular level to the molecular level. And over the years, we've tried to develop our technology and apply it in particular to the problem of phagocytosis. So I'm sure you've all seen images like this. I'll be talking primarily about macrophages. The cell in there is a macrophage, and you can see it has a pretty hearty appetite, and it will lead any number of large beats. They say that phagocytes can eat something bigger than their head. So what's phagocytosis for? Well, most of you are familiar with phagocytosis in the context of the innate immune response. Phagocytes, like macrophages, dendritic cells, microglia, and particularly macrophages, are attracted to recognize, ingest invading microorganisms. And this can be bacteria, they can be yeast, they can be other fungi. So maintaining our body free of infection is the main job of the innate immune response. But there's a second component that we are not so much aware of, and we definitely should be, and I'll try to convince you, and that relates to maintaining the homeostasis of the body. So first, innate immunity. Our macrophages and neutrophils approach invading microorganisms. They ingest them through phagocytosis. They proceed to kill them with a series of very sophisticated microbicidal processes. They degrade the contents of the phagocytosed material. They break it down and present peptides as antigens to instruct the adaptive immune system as well. So there's a connection with the lymphoid system. And because it's an emergency, because you have an infection, the macrophages or neutrophils that first detect an infection attract or try to attract other cells, increase the blood flow, and as a result, they produce inflammation. So the phagocytic response can be inflammatory. But you also have the other component of phagocytosis. That's entirely different, and that's intended to maintain the well-being and homeostasis of the body at rest. And that implies clearing apoptotic cells, removing extracellular debris, remodeling tissue, contributing to morphogenesis, all of those things that are happening in the background, and in fact should not and are not inflammatory. In fact, they're anti-inflammatory. They help with wound healing, and they're quiescent compared to the microbicidal response. Now, Should we care about the responses? Are they important enough? Well, just to give you an idea of the numbers, every one of us turns over every day about 170 billion red cells. Each one of those effete old red cells has to be removed from the circulation by phagocytosis. So each one of you has 170 billion phagocytic events every day. To replace each one of those red cells, an erythropoietic process has to extrude the nucleus from an erythroblast. And that nucleus doesn't go to heaven. That nucleus has to be eliminated. It's eliminated by phagocytosis. To keep the numbers constant, you have 170 billion new red cells generated, 170 billion phagocytic events during erythropoiesis. All taken together, that's 350 billion events. That's more than the federal deficit and then some. But that's not even the entire number. We have neutrophils being exchanged, we have thymocytes undergoing involution, and something we don't think about very much. Every morning when we wake up, our photoreceptors lose a little piece. Cones and rods have a phagocytic event that chews up the part of the photoreceptor that's been used during the previous day. In your brain, when you generate a memory and then have to forget that memory, then the synaptic connection, the synaptic boutons that were formed have to be removed. That removal process is again phagocytosis, and that's undertaken mostly by microglia. So I hope this gives you a perspective of how incredibly frequent and incredibly important this non-inflammatory form of phagocytosis is. Okay. So why is imaging so important? Well, certainly has been for us, because phagocytosis is an extremely dynamic process, as I tried to show in that first movie. The structures that are involved, whether they are dead cells or debris or bacteria, they're in the range that we can resolve by optical microscopy. Everything happens in the course of seconds to minutes. It's extremely convenient to do imaging. It's not something that you have to stay in the lab overnight or waste your weekend looking at it. It all happens within your day's work. It's amenable to use with a variety of fluorescent probes. And they can be recombinant, they can be synthetic, chemical, a large variety. And phagocytic cells are transfectable. So you can put into them all kinds. You can put into them all kinds of transfected probes to study the proteins or the lipids, as you'll see, of your choice. I hope this is working better than the other microphone. Good. So here's a quick example of what you can look at when you use a fluorescent marker to look at phagocytosis. This is a macrophage. It's already ingested a large bead, like the ones I showed you before, and it's about to ingest a second one. Now, the probe here is GFP-labeled actin, and this is just intended to show that phagocytosis is an actin-driven process. You can see how the second particle is being engulfed, and that actin is polymerizing at the leading edge of the phagosome. Okay, now when you're eating a large bead of that size, it's easy to know where your target is and where things are going to happen. When you're eating a little bacterium that is swimming around, you'd be lucky to catch such an event. So if you want to study the molecular details of phagocytosis, you really want to keep all the events in a single defined focal plane. And you cannot depend on the bacteria to cooperate. So what we do instead is we mimic the phagocytic event in a single plane by coating a cover slip with a substrate that the phagocytes find appetizing. So we take the cover slip, coat it in this case with an immunoglobulin that is an indicator of a phagocytic event, and then we parachute the cells onto the cover slip. And then you know that the interaction with the phagocytic target is going to be initiated and remain within a single focal plane. So that's the macrophage spreading on the surface. And now imagine that you are yourselves lying with your face up underneath the cover slip, looking at what's going to happen. And here's actin progressing during the course of phagocytosis. And I told you these guys can open a mouth bigger than their head, and you can see they're spreading, spreading, spreading. And a lot of interesting events happen, all of them, in this case, on the very same focal plane. And as you could see, actin gets completely cleared from the center of the phagocytic cup, and then you get all of these funny punctate structures that are progressing along the development of the phagocytic cup. If you look at those punctate structures in more detail, and everything I've been showing you so far is conventional spinning disk confocal microscopy. If you want to look at things in more exquisite detail and you want higher resolution, you perhaps want to apply STED. So here is frustrated phagocytosis on a single focal plane visualized with STED. And in this case, the cells have been fixed, permeabilized, and stained either for actin or vinculin. And what you can see is there is exquisite colocalization of actin and vinculin. But if you look carefully enough with STED, you'll see that they're close but not together. In fact, the actin is always in the middle of the punctate structure, surrounded almost entirely by vinculin and other components of what are called podosomes. So this is the kind of thing you can do with a higher resolution STED. Now, this is not an artifact of looking at phagocytosis in a single focal plane. We had to convince reviewers, obviously, that this is happening all the time. So here is one phagocytic event that we captured around a three-dimensional particle. You can see the particle in the middle. I'm going to make it disappear so you can appreciate what's around it. There you go. So you can see again a ring of punctate structures at the leading edge of the phagosome where you have the actin in the middle, vinculin around it. Actin is red, vinculin will eventually come in. There you go. Hard to appreciate their colocalization unless you re-superimpose them, which is there. Okay, good. So it's a sophisticated process. You have actin polymerizing, you have actin clearing, you have podosomes forming, you have other proteins coming together with the actin. So how does it all happen? How does it start? Well, it's a receptor-mediated response. And you're all familiar with receptors. For instance, growth factor receptors. The growth factor comes, binds to the receptor, and the receptor activates a tyrosine kinase, or perhaps hormone receptors, G protein coupled receptors. Again, the ligand comes to the membrane, binds to the receptor, activates G proteins. Phagocytosis is more complicated than that. Here, you're lucky that the growth factor and the hormone or neurotransmitter come to the cell. You know, you cannot convince a dead cell to come to the phagocyte. They're really dead. So what you have to do is, like the commercial, reach out and touch someone. So the phagocytes have to get there. Like so. Okay, so how do we get there? How do you reach contact with an immobile target? Well, here's another method that we've employed. This is lattice light sheet microscopy. And again, we're looking at macrophages transfected with GFP actin. And what I hope this will convey is the dynamic nature of macrophages at rest. These cells have not been confronted with any phagocytic targets yet. They're doing the wave all the time. These guys are reaching and extending and looking for possible prey, whether that prey is infectious or a dead cell. Okay. So how do phagocytes compare to other cells? Well, whoops. Nope. Uh-oh. That is not showing for some reason. Well, I was going to show a movie where I would compare a HeLa cell with a macrophage where the macrophages are doing the wave, and the HeLa cell was like watching paint dry. They really don't do very much of anything. So they're doing the wave, they're moving around, but can they exert force? Can you bring the target in? So for that, we've applied yet another method, and these are optical tweezers or laser tweezers. And I'm sure you guys are more familiar than I am with the basis of the technique, but in essence, you use a laser beam to capture a particle and are able to mechanically displace and hold targets with light. So what we've done here is captured with the laser tweezers a red cell and have brought it to the vicinity of a phagocytic cell, a macrophage. We are not touching. We're leaving it, placing it about a couple of microns away from the phagocytic cell and asking the question, can the phagocytic cell reach out and capture that target red cell? And so this is the initial approach. After 60 seconds, we try to retrieve the red cell by moving away the laser trap. And now the laser trap can't compete with the phagocyte. The phagocyte, if you can see, stretches a little filopodium or lamellipodium, grabs onto the red cell and beats out the laser tweezers. So these guys can reach, grab, and don't let go. Okay, so we managed to devise a system where we can capture or at least attach to the particles. Now we have to activate the receptors. Now, unlike the other receptors that I mentioned before, which are activated simply by binding the ligand, phagocytic receptors have to cluster together. A single phagocytic receptor, when engaged, is inactive. You need to cluster them in bunches. So how do we do that? Well, we're all familiar with the fluid mosaic model of membranes that Singer and Nicholson introduced almost 50 years ago, where proteins can readily diffuse in a sea of lipids. So no problem. Once we get there, the receptor's going to move around and eventually are going to cluster. Well, not so easy, not so fast. People like John Heuser or Kuzumi decided to look at the ventral side of the plasma membrane by electron microscopy, and what you see is this incredible mesh of actin, myosin, and other cytoskeletal proteins. So it's not just a fluid mosaic model, it's a fluid mosaic subtended by a very heavy mesh of actin and actin-associated proteins. So that is too simplistic. This is more like the real McCoy. You have the fluid mosaic, but underneath you have a whole bunch of corrals and meshes that are, in principle, retaining proteins and keeping them from diffusing. So how do we find out what's going on? How do we ever get phagocytosis if things cannot move according to Kuzumi? Well, what we did in the lab, and this is Spencer Freeman, a former postdoc, now a colleague, he generated antibodies to phagocytic receptors and attached the antibodies to quantum dots. So with quantum dots, we have enough brightness per particle that we can monitor the behavior of single phagocytic receptors. And to avoid clustering by the antibodies, we use fab fragments only. So single phagocytic receptors, yet another microscopic technique, single particle tracking using either Q dots or chemical fluorophores. So when Spencer measured the mobility of phagocytic receptors, he actually found that the front of the cell and the back of the cell behave differently. Turns out that the back of the cell, as predicted by Kuzumi, immobilizes the receptors. When you see these black dots, it means they're confined. They're moving within a very constraining corral and cannot go too far. But if you look at the front of the cell, you get these lighter blue tracks that are much greater that are indicative of Brownian motion. So in the same cell, the same receptors behave very differently in the front and in the back. So the cell knows its head from its tail, and the cell wants to eat with its head, not with its tail. Why is it different? Because the actin that constrains the receptors in the back of the cell is different from the actin in the front of the cell. And in fact, the actin in the back of the cell is mostly linear, induced by foramins. The actin in the front of the cell is mostly branched, induced by ARP2/3. So you learn a great deal about the biology of what the cell is doing and wants to do from looking microscopically at the behavior of single receptor proteins. If you look now at the efficiency of the mobility of these receptors, you find that bacteria, which are shown here in red, end up being captured at the front of the cell and not at the back of the cell. And this is a result of the ability of the receptors to cluster, plus the fact that the front of the cell shows much more of this ruffling that I indicated before. So the polarity of the cell is tailored to approach, bind, and activate the receptors. Okay, good. We've gotten to the stage where at least we reached out, touched the target, and managed to cluster the receptors because they have elevated mobility in the front of the cell. So now what? Well, now you have to activate kinases, and in particular, Sarc family kinases. And that, in principle, sounds straightforward, but nothing is ever straightforward. It turns out that Sarc family kinases are normally inactive, and you have to activate them first. No problem. The inactivation of the Sarc family kinases is caused by phosphorylation. So you have to dephosphorylate them. No problem. You have phosphatases like CD45 or CD148 that dephosphorylate and activate the Sarc family kinases. So now you can cluster them in the sites of phagocytosis. The problem is when active, Sarc family kinases work by autophosphorylation. And that autophosphorylation can be reversed by phosphatases. And it's the very same phosphatase that activates that also inactivates. So now we have a real conundrum, right? We want to have our cake and eat it. We want to have the phosphatase around to activate the receptor, or the kinase, and we want to remove the stupid phosphatase because now we're activated. We don't want to dephosphorylate it. So how does it work? Well, let's go back to frustrated phagocytosis. Plunge the cells onto the cover slip and watch now the behavior of the phosphatase. This is CD45, the same phosphatase that activates and inactivates. And again, what Spencer did here was to tag with a quantum dot the extracellular domain of CD45, the activating and deactivating phosphatase. And what he found, and I hope this works, yes, is that, yes, the phosphatase is mobile, but the phosphatase is suddenly all confined to the outside of the cell, outside of the phagocytic rim. And again, if you use the same classification code with dark blue, light blue, you can see that most of the phosphatase is excluded from the area of contact with a particle, and it's mobile. So what's going on here? How do you keep something immobile at the front of the cell from diffusing back into the area of contact? This is very puzzling. Well, the secret lies in the physical size of the phosphatase compared to the size of the phagocytic receptors. Turns out the phagocytic receptors are short, and they bind the particle very tight against the surface of the phagocyte. The phosphatases are long and rigid. What happens when the phagocytic receptors attach to the target is that they squeeze out the phosphatases by virtue of their long extracellular domain and their rigidity. It's like squeezing toothpaste out of the paste tube. And so now that the receptors are engaged, the phosphatases are removed. So in the initial stages, the phosphatases are present, able to activate the receptors. As soon as the receptors get engaged and cross-linked, and you're at danger of dephosphorylating them with the phosphatases, you just squeeze out the phosphatase from the relevant site of contact. Good, so now we have touched the target, clustered the receptors, managed to activate and keep activated the Stark family kinases. So what's next? Now don't panic, I'm not going to take you through this whole very complex series of events, but I want to concentrate on one in particular, which is here, PI3 kinase. And the reason I chose this is because now signaling, which has been an entirely protein-dependent event so far, becomes lipid-mediated. So the PI3 kinase is the kinase that converts PI45P2 into PI345P3. So that's all good. How do we study lipids? How do we know that the kinase is activated and what becomes of the lipids? And as I mentioned before, these cells are transfectable, and over the years, a number of groups have developed probes based on domains of signaling proteins that recognize the head groups of specific lipids. And so there are some domains of proteins that recognize PI3P, others recognize PI45P2, others recognize 345P3, which we're interested in at the moment. And how do you monitor this? Well, you take these domains and make a chimeric construct with a green fluorescent protein. And so you can study the lipid of your choice by transfecting into the cell the probe specific to your lipid prior to the phagocytic engagement. So here's one such experiment. Now, again, macrophages, again, GFP, but now we're not looking at proteins. We're looking at lipid. The probe itself is a protein, but the target is a lipid. If the lipid is not present, as is the case before phagocytosis, the probe has nobody to bind to. It's floating aimlessly in the cytosol. It's not very informative at that stage. But once you give targets and the cell starts generating the lipid of interest, now, as is beginning to occur here, you're forming a phagocytic cup, a phagocytic mouth, and the cell starts locally synthesizing PI345P3. Now, I can talk your head off. I'd rather show you what happens, and this will be a temporal course of what happens to PIP3 during phagocytosis, and I hope you'll appreciate that there are multiple phagocytic events happening in both of these cells. Lots going on. Now, let me take you through what happened, not by replaying the movie, but just by highlighting some of the stages at the end of this particular movie. Here's a phagosome that's beginning to form. You can see the shape of the target. It was a red cell. And you get lots and lots of PIP3 forming locally at the site of phagocytosis, and only there, even though it's a lipid, and even though it's supposed to diffuse along the plane of the membrane, it's exquisitely restricted to the phagocytic cup. How it happens, we still don't know. Once the phagosome seals, that's illustrated in this stage, all the PIP3 that was in the plasma membrane ends up around the membrane of the phagosome. It's an endomembrane now that has the PIP3. And the plasma membrane has none left. It's a remarkable segregation process. This is immediately after the phagosome seals. This is about a minute later, maybe 30 seconds, you see remnants of PIP3, and a minute and a half later, there's no PIP3 to be detected. Okay? It's an incredibly localized and extremely transient phenomenon. You try to look at this by biochemical means, by grinding cells up and doing chromatography, you're up the creek without a paddle. You'll never see this. Imaging is the secret. Okay? Now we can see PIP3, as I showed you before, but we can see other lipids, and we can see multiple lipids simultaneously. So what I'm showing you here is actually in cyan, in blue, PI45P2. That's the precursor lipid of PIP3. And you can see that to begin with, it's present all around the membrane. PIP2 not only is the precursor of PIP3, it's also the substrate for generating diacylglycerol. So PI3 kinase makes PIP3, phospholipase C breaks down PIP2 into diacylglycerol. So in this case, yellow is diacylglycerol. Now, again, I'll take you through what's happening here, and we slow down the process here by using room temperature. So, oops, back. Lots of PIP2 in the membrane to begin with. Here's a phagocytic cup and another one that's beginning to form, highlighted there. Lots of PIP2. Even before the phagocytic cup seals, you start getting disappearance of the blue and appearance of diacylglycerol at the base of the cup. Okay, if you look at the same phagocytic event a couple of minutes later, and this is slowed down by room temperature, you see that the endomembrane that formed, that phagocytic vacuole, is loaded with diacylglycerol but has no PIP2, whereas the membrane that just sealed over top of that phagosome retains PIP2, but there's no diacylglycerol left in it. So again, exquisite segregation of lipids, extremely accurate temporal definition of these events, can only be looked at in live cells, can only be looked at in individual phagosomes, individual cells, and probably can only be looked at by imaging using the right probes at the right time. Okay, now all of these lipids, if you haven't thought about it much, are actually charged. They have phosphates. It turns out that compared to phosphatidylethanolamine or phosphatidylcholine, which are zwitterionic and have no charge, the phosphoinositides, because they have phosphates, sometimes multiple phosphates, are negatively charged, and in fact can be very negatively charged. You can have charges of -4 in something like PIP2 and more in PIP3. And maybe you haven't given this much thought, but certainly gave us some clues and anxiety at the same time. What's happening to the charge of the membrane, and what's the relevance of charge in the membrane? And how the hell does one measure charge on the inside of the membrane of a live cell? So for that, we designed a number of probes, and the concept is very simple. If it's negative charge that we want to measure, let's use a polycation. And that polycation is really polylysine or polyarginine. So we made synthetic peptides that have polylysine or polyarginine. Now, if you just transfect a cell with a very positive peptide, it'll go to the most negative part of the cell. That's the DNA. We don't care about DNA. I've never cared about DNA. So you want it to go to membrane, so you have to direct to positively charged peptide to membranes. And we do that by giving a coincidence second signal. We know membranes are going to have a hydrophobic component. We're looking for negatively charged membranes. And so what we do is we generate probes that have not only polycationic stretches, but also hydrophobic determinants. So we put a myristylation or some other prenylation form at the one end or the other of the molecule, or we make it an amphiphilic helix so that there's a hydrophobic component and a polycationic charged component. So do these things work? Do they go to membrane? Do they go to negative membranes preferentially? Now you may remember that in the resting cell, all the PIP2 was in the plasma membrane, and PIP2 is negatively charged. So we were hoping and anticipating that the most negatively charged membrane in the cell would be the plasma membrane, and I wouldn't be telling you if it didn't work. So here are 3 different probes labeled with a red fluorescent protein. These are all synthetic transfectable constructs that have polycationic stretches, a hydrophobic domain, and the red fluorescent protein all in one single construct. You express them, and they go exquisitely to the inner leaflet of the plasma membrane only. And whether it's myristoylated or whether it's amphiphilic or prenylated, whether you have arginine or lysine, they all go almost exclusively to the plasma membrane. An indication is that the plasma membrane is the most highly negatively charged membrane of the cell. That's good. If you compare that to other organelles, you can see that the endoplasmic reticulum doesn't stain with them. Here now the colors are reversed. Green is the charge, red is the endoplasmic reticulum. The Golgi doesn't have very much negative charge, but the plasma membrane certainly does. Okay, so as we anticipated, the inner leaflet of the plasma membrane, which is rich in PIP2, binds cationic proteins. So what happens during phagocytosis? Back to our favorite phenomenon. Now here we have two colors. Green is just a generic membrane marker. Red is the charge marker. And you can see that both of them label the same compartment. And if anything, the surface charge marker is more specifically labeling the plasma membrane. Now we've fed particles to these cells. They started phagocytosis, and you can already see there are some phagosomes that have formed or are forming. But this is a movie, and I want you to see what happens too. There are phagocytic events here, down here, here. So when the movie stops, which is right around here, come on, stop. Hello? Okay, when the phagocytic event stops, you can see that this is a membrane derived from the plasma membrane because it retains the green, at least for a while. But even at this stage, immediately after sealing and formation of the phagocytic cup, the charge is gone. And that was at the same stage where the PIP2 disappeared. So there are acute changes in the negativity of the plasma membrane during the course of the phagocytic event. Why do we care? Why should we care? Well, it turns out that there's a plethora of biologically highly relevant molecules that are positively charged and whose positive charge targets them to the plasma membrane. And these are molecules that you're all familiar with, like RAS, like RAC, like CDC42, like the Marx proteins, and even some kinases like SARC. These proteins are normally attracted to the plasma membrane because of their charge and the charge of the plasma membrane. The implication is that upon phagocytosis, when the charge of the plasma membrane drops, all of these proteins that are associated with the plasma membrane will fall off. So if you had RAS or RAC active on the plasma membrane, you no longer will have it there. If you're thinking about something like RAC or CDC42, and you think that these proteins need to be activated by GEVs or inactivated by GAPs, well, you don't even need a gap. Those proteins are falling off the membrane because of the change in charge, regardless of the activity of the GAPs. So charge matters. Okay, so I mentioned probes for PIP3, for PIP2. We also have a very effective probe, several of them, in fact, for PI3P. So what happens to PI3P? Well, PI3P is not present in the plasma membrane and is not present during the phagocytic event, but it gets recruited to the phagosome immediately after sealing, within about a minute. And once the PIP3 disappears and the PIP2 disappears, now PI3P gets recruited, and gets recruited massively. Here's one phagosome, here's another one. And this is not such a transient event. This lasts throughout the early stages of maturation of the phagosome. And it coincides with some markers of early endocytic events. And so I guess you get a sense that there is a very elegant and subtle choreography of phosphoinositides. They appear, they disappear, they reappear, other ones come in, and all of this is quintessential to the formation of the phagosome and the evolution of the phagosome from a passive compartment to a microbicidal degradative compartment. All of this is coordinated and orchestrated by changes in the lipids. You start with PI4P and PIP2, you form from PIP2, PIP3, the PIP2 disappears, the PI4P disappears, PI3P comes later at the time when cells or the phagosome has RAP5. Then RAP5 disappears and RAP7 comes in. When RAP7 comes in, you again get PI4P. So lipids come, lipids go, and all of this is because you're recruiting kinases, lipid kinases, you're recruiting lipid phosphatases, and it's all essential and intimate to the process of phagosome formation, phagosome maturation, killing, and degradation. So now just to summarize, you start out with a phagocytic cup. You proceed to an early phagosome that later becomes eventually a phagolysosome. And one of the features of this is a drastic drop in the pH. You go from 7.3, the extracellular pH, to pHs below 5. This is 2 units. That means it's 100 times more acidic, more like 300 times more acidic in the lumen of the phagolysosome than it is in the native nascent phagosome. So how does that happen? Well, the culprit of that acidification and an essential component of the maturation and killing and degradation is the vacuolar VATPase, or proton pump. This is an ATP-driven pump that is acquired by the phagosome, as I'll show you in a second, and starts using cytosolic ATP to drive 3 protons per every molecule of ATP into the lumen of the phagosome to make them and keep them very acidic. So you pickle the targets to death, you degrade them, you provide an optimal environment for hydrolases that include proteases and lipases and glycosidases. So this molecule is quintessential. The VATPase is a multi-subunit protein. It's ubiquitous, and because it's ubiquitous, it's been bloody hard to make really good antibodies, because every cell and every tissue and every organism has one of them, and they're highly homologous. Here's just an example of the fact that you need the VATPases for the degradation of the phagocytic targets. So just to take you quickly through it, red are targets that are ingested by a phagocyte, and if you look 4 hours after phagocytosis, they're gone. They've been degraded, they've been acidified, they've been eliminated. If you inhibit the VATPase with a specific powerful inhibitor, such as concanamycin, you still phagocytose, but if you look 4 hours later, the targets of phagocytosis still remain. So if you want to kill, if you want to digest, if you want to generate antigens, you need the VATPase. So can we look at the VATPase in the course of phagocytosis? And as I mentioned, the protein's highly conserved. The antibodies that you can generate tend to be pretty crummy and expensive. So instead, we collaborated with a colleague in our hospital that was using a bacterial protein that the bacteria themselves use as an inhibitor of the pump. As usual, Mother Nature is smarter than we are, and the bacteria have developed a protein that they inject into the cytosol of the host cells and inhibit the VATPase. The bacteria is Legionella, and the protein is called CytK. And CytK binds to one of the cytosolic subunits of the VATPase. So we collaborated with Dr. Grinstein, made recombinant CytK, and added it to cells to monitor the acquisition of VATPases to the phagocytic vacuole during the course of maturation. And so here we have dual labeling. You can see a nascent phagosome where the membrane is green, and you can see that there's virtually no VATPases on it yet. They're in the neighborhood, they're sniffing it out, but they're not there. If you look at the RAP5 stage, which is the early phagosomal, the early endosomal stage, you start getting some VATPases onto the membrane of the phagosome. But if you wait long enough, if you get to the phagolysosomal stage, when they really have to acidify and go to pH 5 or below, then you really acquire an awful lot of proton pumps. So again, imaging and the right probes are the way to monitor the signaling, the function, the degradation, the recycling of the phagosome. So what's involved in phagocytosis? Well, just about anything you want in biology: ligand receptors, signal transduction, cytoskeleton, generation of reactive oxygen species. All of these things you can measure by imaging. All of these things are really a compendium of cell biology. Just about everything you want to know about cell biology is included in phagocytosis. And how do you look at it? Imaging, imaging, imaging. I don't think I have to sell this to you guys, but you know, I trust that this has given you sort of a bit of a flavor of how flexible and resourceful and powerful imaging can be to study this particular biological problem. Now, I'll take the last 10 seconds to plug in a commercial here. In Toronto, we were finding that there's a lot of cell biology to be done. There's a lot of trainees. Now, my wife and I, came from Mexico, and we know there are a lot of trainees in Latin America and other Spanish and Portuguese-speaking countries who would dearly love to come to Canada or to other countries to learn about imaging, to learn about cell biology, and don't have the resources and don't have the expertise. So we created a special fellowship that supports visiting Latin American, Spanish or Portuguese trainees every year, $50,000 to spend 4 to 6 months in the lab learning about cell biology, learning about imaging. If any of you is interested in this possibility, talk to us, or you can go directly to the website of the Pan American Biochemical Molecular Biology Society. And I think I'll stop here and thank again Claire for inviting me, and I'm merciful for her poor memory for having invited me twice.
Doesn't care about DNA. That was fantastic. Thank you so much for that visual walkthrough of phagocytosis. We'll open up the floor to questions. And so, Elke, if you...
Fantastic talk. Thank you very much. I have to admit that I come from a background of protein structure, and I'm just wondering, when you're tagging these lipids with fluorescent proteins, would that do something to the, like the fluidity, to the, you know, exchange and stuff? Does it affect the cell, because you're doing this while the cell is alive?
That's a really good question, and I'm prepared for it because I've been asked the same question for the last 15 years. Everything that you monitor with a probe, you're by necessity interfering with. The extent to which you interfere depends on a couple of things: the abundance of what you're probing, and the affinity and abundance of what you're probing with. If you have a lipid like PIP2, which is extremely abundant in the cell, all you need is a medium affinity probe to see it very well, and you need to express very little to see it very well. The likelihood of interfering with the biology PIP2 is minimal. If you have a lipid that is very scarce, like PI35P2, you need a very high affinity probe, right? And better express high amounts of the high affinity probe. You have a little too much of that probe, and then you're immediately scavenging away a significant fraction of what you're trying to measure. So it's Heisenberg's uncertainty principle, right? You cannot be measuring the lipid without doing something to the lipid. So I'm not going to claim that it's totally harmless, but I want to take the opportunity to emphasize you have to be careful, right? You don't want to overexpress these things and look at the cells that are brightest because they're easier ones to see. In fact, you take the minimum level of expression that will give you a good signal-to-noise ratio and ignore the really bright cells because you know the likelihood of interference is greater.
Thank you.
Hi, very nice talk. I want to ask you about if you have explored about the biophysics of the membrane at the phagosome, because PIP2 and PIP3 are well known to basically organize there as a sort of raft, and there are biophysical properties that change in there, in particularly in the stress of curvature that PIP2 or PIP3 generate, and also if you have any connection with divalent cation like magnesium and calcium, because they are very much engaged in the final outcome of the PIP2, PIP3.
Right. So again, the distribution of the lipid and the availability of the lipid are critical to the measurement. We tended initially to assume that the lipids were homogeneously distributed, mobile, and single molecules. I think we are more educated now, and we know that the lipids self-assemble into islands or rafts, that they may bind to specific proteins, and that what we're looking at with our probes is a global average of the entire behavior. So we will need in the future higher resolution dynamic probes, hopefully looking at single lipid molecule to see what their behavior is. I think a lot of what I showed you here was done with a very simplistic mindset, thinking, well, lipids are lipids, and let's look at them and see what we can see. But I think there's a lot more subtlety, and I think people realize and recognize that the side chain of the lipids, for instance, even within PIP2, you may have more or fewer saturated side chains, that the same PIP2 may be associated with a raft or maybe in a non-raft. If we're looking at the head group, you're looking at the head group. You're not looking at anything else. So there's a lot of detail and subtlety that we're missing in global measurements, for sure.
Yeah, over here, Sergio. So really nice talk, Sergio. The membrane cytoskeleton was originally inferred from diffusion experiments, and then Kasumi came with these beautiful EM images of the actin cytoskeleton, I think 2007, if I recall. Have you tried to image, fluorescently image, the underlying actin cytoskeleton that Kasumi sees? And if not, what are the problems? What do you think it's going to take to actually image the actin cytoskeleton that restricts membrane diffusion?
Right. So, those images that I showed you of the actin cytoskeleton, that's all, scanning EM and their, prototypic cells where you have lifted the membrane and looked from underneath at a generic area. Again, back to the subtlety issue. I think, we need again to go back in there and look at the specific arrangement of the cytoskeleton over time and not necessarily using EM, because that's not going to give you the resolution that you want. It's an extremely dynamic event. It's determined by a combination of the small GTPases and of the behavior of the anchorage proteins, which may be what you're aiming for. There's a whole variety of proteins that anchor that actin cytoskeleton at different times in different places, regulated by different things. So you have the ERM proteins, esrin, rhodexin, myosin, but you can also have myosin 1, and you can have a variety of others that are either more stably or dynamically altering these things. And all those interactions are themselves regulated by phosphorylation, by lipids, for instance, esrin, rhodexin, myosin, which interact with the cytoskeleton, are attracted and maintained at the membrane in part by interacting with the head group of PIP2. So if you think about the hydrolysis of PIP2 during the course of phagocytosis, esrin, rhodexin, and myosin that were attracted that way fall off and the cytoskeleton falls off. What determines the subtleties of the dissociation and remodeling of the cytoskeleton? Again, it's very preliminary stages.
Brandon?
Excellent. Thanks for the great talk. I was curious, the polycation probes that you're using for membrane charge, can you kind of explain the discrepancy that there kind of seemed to be when you showed that there was PIP3 on the closed phagosome that went away after a minute or two, and then in the charged one where it looked like immediately upon closure it was completely devoid on there?
So again, what's the specific question?
So PIP3 should be the most highly negatively charged of the PIPs, but then it looked like the charged one was never on the closed phagosome at all.
So the charge on the probe remains there and is constant, right?
Right. Yep. But I guess my question is, it seems like upon closure, you should see it there because you have PIP3 there.
So upon closure of the phagosome, the charge probe falls off right away.
Right.
That's because the high level of charge of the plasma membrane drops immediately because of the hydrolysis of PIP2.
Even though you have PIP3 on the closed phagosome.
Say that again?
Even though you have PIP3 on the closed phagosome?
PIP3, yeah, sure. Very good point, and I'm glad you bring it up. Again, and that refers to the very first question that I was asked. What probes you're measuring and what's the abundance of a lipid? So the total amount of PIP2, and it's a very good question, the total amount of PIP2 that's converted to PIP3 is tiny. Okay, the probe shows the PIP3 beautifully because it's a very sensitive probe. The amount of PIP2 that gets hydrolyzed is much greater than the amount of PIP3 that is formed. Even though the probes may deceive you into thinking there's a lot of PIP3. You were right. If all the PIP2 were converted to PIP3, the probe should accumulate rather than fall off. The reason it doesn't is most of the PIP2 is hydrolyzed by phospholipase C. It loses the head group altogether.
Okay. Excellent. Thank you.
Matthias?
Yeah. Thank you very much for the talk. It was very inspiring to see how you leverage all of these imaging techniques to answer your questions, to investigate your questions. So my background is a bit more in neuroscience, and we are more and more requested to validate the mechanisms in vivo and in more like in vivo settings to see if we can replicate what we saw in vitro in vivo. So my question would be, all of these experiments, I think, correct me if I missed this, it's all plated and it's in 2D. So I was wondering if there are like any new experiment, new technology that tries to look at these mechanisms, but like in a more 3D settings, like either in organotypic slides or like in organoids to see if like the mechanism that you saw also replicates in 3D, because it's not just also the 3D concept, it's like more busy in the sense of like there are like lots of cells and processes that are around, so maybe like less space for the cells to move.
Right. No, I think the potential for using lattice light sheet microscopy, for instance, to do long-term three-dimensional measurement at varying levels of resolution will increase the amount of knowledge that we can gain. I think it's preliminary times. Now, like, I don't know what it's like in your individual labs in your individual countries. Up until recently, there were not too many sources of lattice light sheet microscopes. They're not cheap, as you know. No, we were, I think, one of the first ones in Canada to get ours, and I think Amra Sarich, who's sitting around there, has used it the most. But she can tell you the enormous potential of using intravital type of microscopy, zebrafish lattice light sheet microscopy, Drosophila lattice light sheet microscopy, and possibly other newer techniques to apply all of these kinds of concepts and all of these kinds of probes, in a more meaningful three-dimensional things. Some people will advocate for organoids and measuring these things in organoids. I'm not a big fan of organoids, but no, we do have enough real organisms, small organisms, and even in mammalian cells, people can put the liver onto a cover slip and go ahead and do your measurements. And I know people are doing it now with intravital.
Yeah, just to comment on the latest slide sheet. So I'm at UBC, and we acquired also one of the few latest slide sheets in Canada. And I can say that, yes, it's a very powerful tool to leverage like this very big tissue or like this mechanism that we want to target in 3D. Like, so in our case, in neuroscience, looking at all of the processes in 3D. So yeah, I think it's like a very, very powerful tool to look at such mechanism in the future.
Yeah. Thank you so much for the talk. I appreciate using different imaging protocols. I'm just thinking that because we do EM in the lab, electron microscopy, and then when we image in brain tissue, we see a lot of lysosome, primary lysosome, secondary, tertiary, with different lucency, like different colors. So I'm just like thinking from your talk that could it be that the dynamics of the PIP3 from the initial state to the maturation, could they contribute to these color changes that we see when we image under electron microscopy, when we see lysosomes and endosomes with different colors? Could it be that the dynamics of the PIP, of phospholipids, change? Could it be? Did you do anything to like check on that?
I think no. Any stage of development of the endosomes and the lysosomes, and in fact even the secretory pathway, seems to involve changes in the lipid composition, for sure. You know, the changes in color that you're referring to surely are associated with the maturation stage and the content and so on. And all of that is directed in one way or another, not entirely, but in part, by the lipid composition plus other signaling events. So the answer is quite possibly yes.
Thank you.
Gwen?
We have a virtual question, or a question from our virtual audience. Does phagocytosis play a role in clearing microplastics from our bodies? And can they be degraded in phagolysosomes?
Let me see if I understand correctly.
Yes, the question is also posted up there on the screen, right there.
You're asking me whether the phagosomes always end up as?
Does phagocytosis play a role in clearing microplastics?
Play a role in clearing microplastics from our bodies. Yes, the answer is yes. Phagocytosis will, these guys have an enormous appetite and they'll take up anything that you feed them, microplastics included. They eat, the beads that I was showing at the very beginning of the first movie, that was actually latex. Right now, there's no receptor for latex, but these cells have a variety of receptors that identify foreign surfaces, including microplastics. Now, whether microplastics can be degraded by phagolysosomes, I don't know. I know that when we use latex beads or when we use other types of foreign materials, that the cells didn't evolve to degrade. You don't have the enzymes, you don't have the means of doing it, then they're in all likelihood not going to be degraded. There are a couple of different processes involved in the degradation and elimination. One of them is hydrolytic enzymes. The other one is the effect of pH itself. I don't know enough about microplastics to know if they're pH sensitive. I doubt it. There's also reactive oxygen species. So if the microplastics are broken down by superoxide or hydrogen peroxide, all those things, there's a chance that you may be clearing at least some of it, although that's a transient stage. But to the extent that Mother Nature wasn't counting on microplastics, I don't think we've devised ways of eliminating. Now, thirdly, when cells get tired of eating stuff that they don't like and they can't digest, they just barf it out. They let it go. So there's papers, not too many, with a process they call vomocytosis, like the cells vomit out the particles that they ingested. That wouldn't be very good in the end to clear microplastics because they go back into the circulation. There's typical examples of things that the macrophages can take in and are unable to dispose of, and they'll last for years. And the one I love to quote is tattoo ink, right? When you get injected with the ink of the tattoo, first thing that happens, your macrophages go and eat it, and you still have your tattoo 20 years later, unfortunately, or fortunately.
Thank you so much, and thank you for the wonderful talk.
Yep. Please.
Thanks for the talk. It's very interesting. I have a question on the Fc gamma, the phagocytotic receptor phosphorylation. So in that slide, you talked that these phagocytic receptors can cluster, and then they squeeze out the phosphatase. So that sounds like a phase separation behavior here. And later on, you brought in the PIPs. So I wonder if you have put together of these two events and looked whether there's a phase separation happening there, and also if the membrane lipids is playing a role in regulating them. So you mentioned the molecular size and all, and I understand that. So I wanted to see if you have looked at the lipid role in that.
Yeah. So obviously, what I presented here is the tip of the iceberg, and the regulation of the receptors for phagocytosis is more complicated than I pictured here. The receptors are engaged, are activated, and then shortly thereafter, they have to be deactivated. As soon as the phagosome seals and you get your process to the next stages, the deactivation of the receptors is, again, through phosphatases, but other phosphatases. And as you know as well as I do, or better, you know, there's a whole variety of phosphatases. In this case, these particular phagocytic receptors are tyrosine phosphorylated, and they're tyrosine dephosphorylated. But phagocytosis is not phagocytosis, because phagocytosis of dead cells goes through a different type of receptor. Phagocytosis of particles like microplastics or latex is yet a different types. There's probably several dozen phagocytic receptors. And the other compounding factor, compounding in the big scheme of things, but not for us, is that when we study phagocytosis and we do frustrated phagocytosis on glass, we put a single purified ligand that will engage a single purified or single type of receptor, because we want to simplify our lives and understand what's going on. If you take a bacterium or a fungus and you feed it to a cell, you engage probably half a dozen or a dozen receptors. If you give a dead cell, you do the same thing. You engage a whole bunch of other receptors. And as you would have predicted, the receptors you engage when eating a dead cell or debris are very different from the other ones. These ones have to be inflammatory. The other ones have to be anti-inflammatory. So, take the example of the Fc gamma receptor phosphorylation, CD45, as one simplified single instance of what's happened in phagocytosis in a model system, not even in reality.
Okay, thank you.
Hi, another question from our virtual audience. This one's more imaging focused. What future directions do you envision for using super resolution techniques to image phagocytic events?
Nobody gives me a break. I'm trying to become emeritus, and you keep asking me for future experiments. Yeah, I mean, there's tons and tons to be done for super resolution. I know I show that very simplified image of the signaling complex that forms at the time of phagocytosis. We really don't know the time course, the number of components, the physical organization of those components, how they get organized and reorganized and disorganized. And that's just at the stage of forming of the phagosome. Later stages, you have to get the maturation, like when you go from a plasma membrane to an early endosome thing, it's not just the same membrane. The membrane is undergoing fusion and fission events, and yet it keeps the same area in the same volume. So how does that work? You have to have fusion molecules bringing things together, allowing fusion to occur on one end with specific targets, and you're having fission occurring somewhere else in the same phagosome, removing pretty much the same area and the same volume to maintain what you have, and generate a second stage, early and late, and phagolysosome that are maturing and changing their personality and changing their composition, and we know diddly squat about all of that, right? And all of that will be done probably using super resolution with the right probes, with the right colors, with the right timing. And again, it would be virtually impossible to do any of that using biochemical means because the system is asynchronous and because the system is transient.
Great, thank you.
We have time and bandwidth for one more question.
Okay. Because
So-
there's one more that's here. We were hoping to use the force for the laser tweezers. We never did. It should be doable, but I know there are other groups that have utilized laser tweezers or have used atomic force microscopy, gluing particles to the tip of the atomic microscope probe, and then expose that to a phagocyte and try to measure the forces involved in pulling and pushing and so on. So again, there's a ton of biochemistry, but there's also a ton of physics and biophysics to be learned using optical and related techniques with phagocytosis and related processes.
Fantastic. We'll have one more question from a live audience member.
So back to your introduction, you mentioned that 1.7, 1.6 billion the phagocytosis occurs in our body. I assume you refer that as to a young person, not old person like me. So my question is, is phagocytosis related to aging? And if so, how are you going to study this using an in vitro system?
So is phagocytosis related to?
Aging?
The answer is I don't know. But we can talk about it for sure.
Fantastic. So lots of questions about the biochemistry, about the methods, and I think lots to talk about during the break. So let's thank Professor Grinstein.